In this video, we'll be looking at the synapsis of chromosomes in meiosis. Synapsis is the pairing of chromosomes in meiosis that prepares them for splitting, and it allows for crossing over of the genetic material, which may lead to genetic diversity. So here we have the first individual with these chromosomes, and the Twizzler pieces will represent the segments of the chromosomes that must pair together in synapsis. So here we have a normal chromosome here with the normal segments, but this one we have an inversion, is that this segment here was taken out, flipped 28 degrees, and put it back in. And it's paracentric because it includes this centromere here. And so next we'll look at how the chromosomes pair together during synapsis. During synapsis of these two chromosomes, there's going to be something called an inversion loop created due to the inversion of the chromosome, so that these all will pair up. It has to create this little structure here, and then once they split, there's going to be chromatids formed. But since there's a crossover between J and K, it'll affect the gametes differently, and it'll make them viable and not viable. So to create these chromatids, they're going to have to be split. These two chromosomes and the chromatids are going to have to be split. So we're going to go from G, H, K, but since it's on the inside here, it's not going to be affected by this crossover, so it's G, H, K, J, centromere, I, L, M. It's going to be chromatid 1, and it's viable because there's no aberrations except for this inversion, but all genes are going to be still present, so it's going to be a viable gamete produced. We're going to have chromatid 2 is going to be on the outside here, G, H, K, but now it's affected by this crossover, so it's going to be G, H, K, J, centromere, I, H, G, which produces a non-viable gamete because it's missing these chromosomes over here, or these genes, sorry. Chromatid 3 is going to be formed by here. It's going to be G, H, I, centromere J. It's not affected by this crossover, J, K, L, M. It's going to be viable gametes produced because it has all the genes present. And then to create chromatid 4, we're going to start from over here. It's going to be M, L, K, on the inside, sorry, M, L, K, crossover, J, centromere, I, L, M. And that's going to be a non-viable gamete because these genes are not present in that chromatid. Now we have individual 2, and as you'll see, there's 2 paracentric conversions this time. And so that means both these segments were taken out, flipped 180 degrees, and put back in. But you'll see it affects the synapsis, and it also affects the gametes differently than individual 1. Here is individual 2 in synapsis. And you'll see that since there's both a paracentric inversion in both chromosomes, they actually pair up quite nicely. And that'll mean that all the gametes are going to be viable because of this double inversion. Now making these chromatids is going to be a lot simpler, so we can just go down the line here for both of these, G, H, K, J, I, L, M, and then G, H, K, J, centromere, I, L, M. Those are not going to be affected by this crossover, so that'll be chromatid 2 and 4. And then on the inside we have G, H, K, crossover, J, centromere, I, L, M. Then the other one, G, H, K, crossover, J, centromere, I, L, M. So you can see that here with these two differently shaded. It just comes from the different chromatids from that crossover. But all these gametes are going to be viable because there's no actual missing genes in those gametes. Individual 2 is more fit because it has all viable gametes.